Okay, we're going to go ahead and get started. We are at the starting time. So thanks for joining us. I'm Tim Anderson, one of the senior research analysts at Bank of America. And this is a 40-minute fireside chat focused on current and emerging treatments for Alzheimer's disease, where, you know, after decades of not having a lot, we finally have some approved products, and we've still got things in the pipeline percolating along, and all feels very exciting. I'll be talking with two of Mass General Brigham's best experts in the area, who are really absolutely world-class leaders in this space. We have Dr. Dennis Selkoe, who's the Vincent and Stella Coates Professor of Neurologic Diseases at Harvard Medical School and co-director of the Ann Romney Center for Neurologic Disease at the Brigham. And we have Dr. Rudy Tanzi, who's director of the Henry and Allison McCants Center for Brain Health, director of the Genetics and Aging Research Institute, and co-director of Mass General Institute for Neurodegenerative Disease, and also serves as the Joseph P. and Rose F. Kennedy Professor of Neurology at Harvard Medical School. These guys have contributed so much in things like the amyloid hypothesis and the underlying genetics of Alzheimer's disease. So it's a great pleasure to have you on stage with us. I thought we would start, talking about current practice dynamics with what's been approved in the last few years, and then we can dig into some of the things that are in the pipeline and how the understanding is evolving and how to target these patients. So let's just start with the amyloid hypothesis. There was a lot of debate, I think, over the years. Now that we have Aduhelm that was supplanted by Leqembi, and we've got Kisunla on the market now, I'm assuming any sort of criticism or question marks around that have been put to rest. Is that correct?
You would think. No, I think we always say now, let's not call it the amyloid hypothesis anymore. It's maybe the amyloid theory or the amyloid posit. But, when Dennis and I first met, and Dennis was a junior faculty member, this was 40 years ago, when I was a student in the neurobiology program at Harvard, he graded my first Alzheimer paper. And at that time, neurobiology of disease, they write the diseases on the board, and everybody picks a disease, and alphabetically, I went last, and nobody picked Alzheimer's. And I picked Alzheimer's, and Dennis was my grader, and he gave me an A-. And it's because in one of my figures, we didn't know, this was 1985 or so we didn't have the amyloid gene yet. But I have to say that what he criticized me about, he was right. So it's okay, I'll take the A-.
So I think that this gradually declining notion that amyloid isn't a worthy target or is a secondary or tertiary event in Alzheimer's should be put to rest. It never made a lot of sense, and if you know the history of this field, you know that George Glenner pioneered the notion that and discovered the amyloid beta protein and felt that this was a organ-limited amyloidosis in the brain. And there's just lots and lots of scientific evidence, especially genetics, but genotype to phenotype relationships that indicate that A beta accumulation initiates the disease. Now, we obviously don't know the molecular reasons why these millions and millions of people who get Alzheimer's, why each of them has a problem with A beta metabolism. We know some of the reasons, and ApoE4 looms large there because it seems to allow less clearance of A beta once it's produced. But we don't know for many people what triggers A beta dyshomeostasis. But there's little or no doubt, I think, at this point, that A beta elevation in the brain leads to tau alteration. It's been beautifully studied by our colleagues, Keith Johnson and his colleagues at MGH. So it looks like until you start altering tau secondary to A beta accumulation, you don't get a lot of evidence of dysfunction, neuronal dysfunction and gliosis. But I think at this point, there are few folks in the field who are familiar with the biology that would question the amyloid hypothesis.
Great. Okay, so let's talk about the real-world experience with the products that we have, maybe just with efficacy to begin with. I don't think anyone's really arguing that these are magic bullets. We know they're not curative. They slow down disease progression. They appear to be disease modifying. I guess a couple of questions within that. Is there substantial variability of response among patients? And then the prescriber views, not just you guys, but the broader prescriber community, what's the embrace of these products and really the push of these products? Is the demand physicians pushing patients to go on? Is it patients asking to go on therapy? What's your experience?
So at Mass General Brigham, we're north of 600 patients that we've put on monoclonals that lower amyloid, i.e. either lecanemab, Leqembi, or donanemab, Kisunla. And the problem in the field has shifted from skepticism about amyloid being an appropriate target, I think that's largely declined, as we mentioned, to are these treatments anywhere near good enough to warrant my family member going on them? And we're finding that a great many people, once they learn about what is required to get into treatment, namely MCI or mild AD, so not very severe clinical symptoms, but you have to have clinical impairment. We're not yet at the point of prevention. We're hoping for that. I think they more and more accept this, but the uptake has been gradual and slow. Partly maybe because of the original Sturm und Drang around the amyloid hypothesis, but also because it requires infusions every 2 weeks or every 4 weeks. And I think a major game changer now is FDA's approval just 10 days ago of Eisai's Leqembi being given subcutaneously after treatment with IV Leqembi first. And they have a rolling application to go to initiate treatment with sub-Q. So that is going to change things a lot, making it much more convenient, much less problematic, although we'll still have challenges. So I think we need even more efficacious agents and agents that are even easier to administer.
I think there's also the safety issues. When folks talk to me about whether they should try monoclonals, they're worried about safety. If they're ApoE4 homozygotes, they're particularly at risk for the side effects of ARIA, meaning hemorrhage or microhemorrhage or vasogenic edema or swelling. I think this is a great step forward to have drugs that clear amyloid. But I think at the end of the day, we're going to need much safer and more affordable alternatives to democratize early detection. We now have blood tests that are approved, that can tell you if you have amyloid and tangles in your brain. And early detection needs to be followed by early intervention. And very expensive monoclonal antibodies with safety issues, are a great beginning, but they're a start. And they're providing proof of concept that the faster you make amyloid, the sooner you get Alzheimer's disease. But amyloid is very much like cholesterol and heart disease. It's something you want to manage early in life through primary or secondary prevention to avoid already going down that road to neuroinflammation and cell death where you have to try to reverse the course of the disease. So we need to get to drugs that are safe enough and affordable enough that they can be used even before early stage treatment, but primary or secondary prevention following early detection, similar to the course of cholesterol, heart disease, and managing that disease.
So let's stay on the topic of safety. Clinical trials, they've routinely shown at least imaging abnormalities in the form of ARIA-E, which is edema, and ARIA-H, which are microbleeds that are almost always, subclinical. But some patients have side effects, especially from ARIA-E. My concern has been, and I guess it still is, as these roll out broadly into the community and you're potentially not managing patients closely enough with imaging follow-up or clinical follow-up, could you somehow get upside down, someone have a bleeding event, for example, maybe it's a patient that has cerebral amyloid angiopathy, and then spook the market. We haven't seen it, and I hope we don't see it, but if you could just talk about the clinical view, what physicians are viewing, are they growing more comfortable with ARIA-E and H? And do you think that patients are in fact being managed well enough in the community? I know it's probably hard for you to answer that last part.
So I think you're absolutely right. We live in fear of some negative event. In our place where we've gotten north of 600 patients, we do follow them very closely, and as soon as we see symptomatic ARIA-E or symptomatic ARIA-H, we stop dosing. We pause dosing for 2 or 3 months until the MRI clears. And depending on the severity radiographically, if it's moderately severe ARIA-E, we will actually tell the patient we don't think it's wise to continue. We will not provide the medicine anymore. So it turns out that any number of E4 homozygotes that I've treated and E4 heterozygotes, which together comprise a large part of our treatment population, can be managed very effectively. But of course, if you don't pay attention to the MRI, if you don't get them on schedule, then like any other potent agent, you're going to run in trouble. Now, we can jump ahead to what lies not so far away, and that is the experience with trontinemab from Roche, which looks like it has a very substantially lower rate of ARIA. So lecanemab, which is right now the most prescribed medicine, was the first one FDA approved, cross genotypes about 13% ARIA, but only a quarter of those, so about 3 or 4% are symptomatic, as you said. So in the lecanemab population, north of 90% of patients who take it will never have a symptom, even if they develop radiographic ARIA. And with trontinemab, the hope is, it's in phase 3, we'll see what happens, that you'll lower amyloid quite successfully, but not with as much ARIA. And that's presumably because of the range shuttle or transferrin receptor targeted bifunctional molecule.
There are also companies now targeting more the amyloid beta oligomers, the dimers, trimers, hexamers that Dennis and I both worked on. It turns out that if you target oligomer and not fibril, there's a greater chance you won't hit the congophilic amyloid angiopathy or blood vessel amyloid that causes the inflammation around the vessels that leads to ARIA. So keeping track of some of those companies as well, if they can achieve the same goals in the brain of clearing amyloid, but avoid the interaction with vessel amyloid, that'll be a big step forward for safety.
And I think Rudy makes a good point, because probably the difference in ARIA-E rates between lecanemab and donanemab is explained by lecanemab putatively more favoring small oligomers. So that's what the company says, of course. We study these antibodies in our lab, and they can clearly bind fibrillary beta also, and they clear it. But lecanemab has a bit of a propensity to bind oligomers, and it does have about half the ARIA rate.
Yeah. Yeah, okay, so that's a great segue, because I wanted to ask, how do you choose between Leqembi and Kisunla, or lecanemab and donanemab? It seems like it's almost equally split in the market when I look at the data. It still might be favoring Leqembi at the moment. And the companies build these differently, and you just touched on that, right? So lecanemab hits not only soluble A-beta, but deposited plaque. And then donanemab only hits the latter. There's a big point of differentiation with the Lilly drug, which is finite dosing, right? You give it until plaque's gone, maybe that takes a year, and then you can stop therapy. I would imagine payers love that. What do patients want? And what do you view as, how do you decide? Are you using both products? And if so, who gets one product versus the other?
So in my experience in our practice, which involves many neurologists at the Brigham and the MGH, we're using both products. We started out, of course, with lecanemab, that was first available. And when you quote the ARIA rates to patients and their families, which of course we do, and explain in detail what we think are the pluses and minuses of these, I think patients, many patients are very concerned about ARIA potentially being symptomatic and therefore favor the agent that has the lower rate. On the other hand, the convenience factor, until recently at least, has favored donanemab, so once a month rather than every 2 weeks, and it is a big burden for people to come every 2 weeks. Now with FDA approving lecanemab maintenance therapy subcutaneously, there probably will not be as much of an advantage to donanemab. Of course, donanemab is also going to be followed by a subcutaneous version as well. So my view is that there's a bit of favorability towards lecanemab versus donanemab right now. And one of the points you mentioned, Tim, is the one-year treatment idea. So Lilly touts the fact that their antigen, pyroglutamate A-beta, will be gone, presumably after 12 months of monthly treatment. And therefore, a clinician and a patient can ask, does it make sense to continue to take donanemab when the target is gone? And of course, the advantage Lilly offers is you can stop. But many of us do not feel that stopping A-beta lowering is necessarily wise. I don't even know that dropping amyloid PET scans to 11 centiloids or less guarantees that there is no amyloid. And I'm sure there is still A-beta oligomers which can't be seen at all by amyloid PET scans, so you don't know what happened there. We're developing a CSF assay and ultimately a plasma assay to measure oligomers before and after treatment. So I would say that donanemab has a more complicated therapeutic plan. You treat for a year, what do you do next? Then you probably pause, stop, and potentially offer the patient to go to another agent. And so I think that makes it a little bit more complicated. And we are seeing patients switch after treatment.
When I dug into the data a number of years ago, or a few years ago, it seemed to me like the data is fairly thin that says soluble A-beta is neurotoxic in terms of actually clinical data. It comes from animal studies and in vitro studies. Is there a debate in the academic community among those that know the space well whether soluble A-beta actually plays an important role or not? Because Biogen Eastside will tell you one thing, Lilly will tell you another.
I would say in both the animal models and more recently in our human mini-brain organoid models, where we're using human cells in a mini organoid, that it's the A-beta oligomers. We call it soluble, really. It's oligomers of A-beta that are pre-fibrillar. They haven't formed amyloid plaque yet. And they're the ones, when they have access to neurons, are inducing tangle formation. So if you look at the familial early onset mutations in APP, the presenilins, the genes we found years ago, the most dangerous ones are the ones that increase the A-beta 42 to 40 ratio the most, A-beta 42 being more prone to amyloid formation and oligomers. So in our model systems, in the organoid models, as we put in mutations that increase that ratio of the long A-beta 42 to 40, you get more tangles forming in response. Once you have fibrillar A-beta, you're not out of the woods because microglial cells are trying to clear the amyloid plaque. And they're choking literally on fibrillar A-beta, and that will cause microglial cells to convert to a neuroinflammatory state and start inducing astrogliosis inflammation, which is causing much more cell death than the original tangles. So the fibrils are dangerous in their own way, I think, for inducing neuroinflammation through microglial activation, astrogliosis. But in terms of the early stage of the disease, I like to say A-beta oligomers are the match and tangles are the brush fires that spread and propagate. But neuroinflammation is the wildfire that takes you out. And the problem I have with the current course of therapy is we're treating too late. You know, immunotherapies are great. They're better than having nothing, and they do clear amyloid, and they help stabilize the disease. But we really need to have agents where we can go in and blow out that match early, get rid of the amyloid, stop the tangles from propagating. Because once you get to the point of major degrees of neuroinflammation, the cell death, the synapse loss is so massive that simply blowing out the match or stomping out the brush fires or tangles is a little too late. So our lab is focusing mainly on coming up with affordable, safe alternatives so they can be used early enough for preferably primary or secondary prevention.
I think another answer to that, I agree with what Rudy says, is that it is very likely that small assemblies, oligomers, have a role to play. If you look for clinical evidence, it would be rare mutations in APP that don't populate fibrillar amyloid very much, but still make oligomers, and those patients have a devastating course of Alzheimer's, like the Osaka mutation. They're very rare, but they create the principle that you don't have to go to full-blown fibrillar deposits to get trouble. And there's a ton of preclinical evidence. It's sort of like re-adjudicating the amyloid hypothesis. We had so much preclinical evidence that it looked like it was right, and then we got clinical validation. And the idea that oligomers wouldn't play a role is very low. You can apply oligomers harvested from patients who died with Alzheimer's disease and treat them very carefully in non-denaturing ways, put them on neurons, and as Rudy's just said, the neurons accumulate tau, the microglia become inflamed, and you can put in lecanemab into the mixture and erase all that. And interestingly, if you lower tau with an antisense oligonucleotide, as Biogen, for example, is doing, and Novartis is doing now, studying, that protects from the neuronal abnormality induced by the OA beta. And I'll mention one other point about treatment now. We have more and more evidence that with the 2 FDA-approved agents, and lecanemab now I think is approved in 48 countries, just as Rudy emphasized, if you treat early, and right now we're not using in the clinic prevention mode. That's being done in the AHEAD study and Lilly's prevention study. That's great. But for people who already have symptomatic Alzheimer's, if they're very mild, if a tau PET scan would, example, show very little tau tangle formation beyond the medial temporal lobe, if their amyloid level is abnormal, but it's below 60 centiloids, that's all been studied as part of the Clarity AD trial, and those people can really become completely stable for 36 months. So there's evidence now, since the advent of these 2 agents, that if you treat really early in symptoms, the people will do remarkably well. And so that's why I think our friends, our relatives, our families, and our patients should come forward and get these treatments. Obviously, it is worrisome that ARIA still is a concern, but perhaps there's an agent coming along that has much less ARIA.
These are all great segues into topics I want to get to, and we will get to them in order. So just in terms of where we are with the kind of the infrastructure adapting to absorb patients coming to therapy, to me it seems like the floodgates might suddenly open here. So the company started, I think, with Leqembi at least, started doing direct-to-consumer advertising in June, so awareness is going to go up. We now have the sub-Q formulation and maintenance, and eventually we'll have it in induction as well. And then we're getting this increasing adoption and understanding of blood-based biomarkers. So where are we in the healthcare infrastructure's ability to absorb patients? How backlogged are patients at MGB, for example?
So I think we're barely ahead of that, and I think it is very important that we gear up the infrastructure. We had initially a waiting time of up to 3 months until the patient, after being seen by one of us, could get infusion for the first time. It's now down to about 4 to 6 weeks, so it's better. We found better infusion capacity. We've gone to Saturdays and rolled up our sleeves and did everything we can to increase infrastructure, but it isn't there yet, and with direct-to-consumer marketing, it's going to really be problematic. So we need more centers, and in Massachusetts, for example, at MGB, we get quite a lot of people from Pittsfield, or we get people from Maine and people from New Hampshire. Rhode Island has some very good treatment centers already. So we're hoping and we're seeing that practitioners, neurologists principally, in less populated centers are offering this agent now. I just recently found a family that I follow, a neurologist in Traverse City, Michigan, that's offering monoclonal treatments, and they hadn't been doing very much of it. Of course, that raises the question, less experienced clinicians are more likely to run into some management problems, but there's also a very heavy load of messaging about how to manage the possibility of ARIA. So we need more centers, and I don't know that our home base, MGB, would ever be able to build up so much that we could take a very large fraction of New England's Alzheimer patients who are MCI mild AD. I think it needs to be happening in many centers and practices, and I think it will.
Yeah. Okay, blood-based biomarkers. We dug into this pretty extensively over the last few years, and really trying to figure out the central question. When are they going to be good enough to completely replace PET scans or CSF testing to confirm amyloid positivity. And it kind of seems to depend who you ask. I think there's proposed specificity and sensitivity criteria out there for when that might be enabled. So are we there yet? And if we're not there yet, when do we get there? Because to me, that's a bottleneck that suddenly goes away in a meaningful way if you could just do a blood test.
I think for purposes of clinical trials, we don't need to do PET scans necessarily, especially if you're on a budget. That initially looking at the A-beta 42 to 40 ratio, seeing that ratio low in a patient indicated that A-beta 42 is getting trapped. It's sticky by plaque and couldn't get out. And if you're clearing plaque, for example, with Leqembi, 42 gets out more and your 42 to 40 ratio goes up in the plasma as an indication of efficacy. But now, given that amyloid is inducing a very specific phospho-tau epitope at p-tau217, I like to say, and Dennis and I have said this a long time, Alzheimer's is an amyloid-induced tauopathy. You could have a head-bang-induced tauopathy like CTE, but in the most common dementia, amyloid induces tauopathy. And p-tau217 is a perfect marker that amyloid is present, and you're starting to make tangles. And so for some of the trials we're doing now of new drugs, and I hope we do talk about upcoming new drugs at some point, we're using p-tau217 as for recruitment without even having to do with really, I mean, you can do A-beta 42 to 40, but p-tau217 covers it. And I'm seeing increasing journal articles that say really the blood tests are really rivaling and benchmarking so well against PET scans that we can use them to judge efficacy of a drug and judge how a patient is doing as they progress through the disease.
I agree. I think the answer to your question is now. I think p-tau217 has an area under the curve against amyloid PET as a marker of 0.98. And there are not too many tests in medicine that get that high an accuracy versus what is currently the gold standard, which would be an amyloid PET. That, of course, itself isn't truly a gold standard. Pathology, at the end of the day, would be, and the amyloid PET won't catch everything anyway. But I think that trialists feel that p-tau217 will allow them to save a lot of money, the sponsors a lot of money in conducting trials. But I think for everyday practice, which is why I'm glad that it's clinically approved, FDA approved, et cetera. And I think you wouldn't want to run out and get a p-tau217. This has to be done in the context of a neurological evaluation because we're not treating pre-symptomatic individuals. Assuming everyone here is pre-symptomatic in this room, getting your PT17 isn't going to allow you to get the medicine. So that has to be done in a medical setting with a
And especially since there are big diurnal changes, and the p-tau217 in the morning versus the afternoon is very different. And so you really need to control how that's monitored.
And really, elevated p-tau217 would only allow you to get treatment if you have mild symptomatic. We're hoping very mild symptomatic, but we will test you and find out how you're doing cognitively. So I think the blood biomarkers are here and can be substituted for amyloid PET. Now, the complexity is that the amyloid PET is very useful to judge outcome because it's unequivocal these antibodies clear amyloid, even if we debate how much they help people who are already symptomatic. So we've seen that we started out in our treatment program at MGB with quite a lot of people qualifying with CSF biomarkers, which also work, so as compared to plasma. But we've now left that aside because those who had CSF biomarkers at the beginning and got on treatment didn't have an amyloid PET scan. There was no reason to do both. And now they lack an amyloid PET scan as a comparator when they go for 18 months of treatment. So in some ways, having an amyloid PET scan, which is we're finding CMS, Medicare, is allowing us to do those, that can be done once at the beginning and once at 18 months or so. Possibly earlier than even at 12 months. And I think that's going to be expensive, but the blood tests can supplant the need to do multiple amyloid PET scans, and certainly for trials.
Yeah. Okay, let's move on to things in the pipeline. So you mentioned trontinemab. This is the Roche compound. The brain shuttle gantenerumab, as they sometimes call it. early data showed very impressive plaque clearance and low ARIA rates, and the product gapped into 2 phase 3 trials, I believe. What do you think about that data? It looks to us striking, and there's a biology behind it that makes sense. How promising is that compound?
I personally think it's quite promising because it probably suffuses into the brain via a capillary route. Endothelial cells are full of transferrin receptor, and the bifunctional molecule allows them to bind to that. And so it is coming in probably not where there's such a heavy burden of amyloid and blood vessel walls, as would be the case with a naked antibody. So ironically, donanemab did well in clearing amyloid by itself, but didn't quite achieve its endpoints. But together with a delivery mechanism that goes to the capillary bed, it seems to work quite well. So, you know, obviously everything is in phase 3 data, and things sometimes turn upside down. But if biology has any rules, this agent looks like it can clear amyloid very effectively, maybe with less frequent dosing, maybe not even dosing it forever and stopping at a certain point and then reintroducing the agent. And so far, the ARIA rates are low, and that would be highly welcome. I also hope that we'll have a little chance to talk about non-biologic approaches. There are other biologic approaches which are very important. Antisense oligonucleotide for tau. I personally like that a lot, to combine with an amyloid-lowering antibody. We also should talk about GSMs, a topic that we both are very favored.
Well, why don't you just go ahead and talk about that now?
Yeah,
GSMs.
I think that, you know, I mean, monoclonals are great, but they're expensive, and they have side effects, and they're getting better. But I think we have to look to a future of drugs that are safe enough and affordable enough to democratize early detection, early intervention, where we're stuck on early treatment when it's frankly still too late. I mean, we're helping to stabilize the disease by removing amyloid, but we need to do much better. This is like, if it was cholesterol and heart disease, somebody has severe coronary artery disease, and you're saying, well, now we're lowering your cholesterol, and we're going to help stabilize new plaque. But let's think more forward about the future of drugs that would be safe enough and affordable enough to use across millions of people who are going to find out with a blood test. Maybe 10 million or more might find out. They don't have Alzheimer's yet, but a blood test says that p-tau217 is high, and they're on the way. So, you know, having a little white pill that does the same thing will be very important. And back 20 years ago, my late colleague, Steve Wagner, who died due to COVID-related problems, we invented a drug at that time. This followed up on work on NSAIDs. It was shown that some NSAIDs could decrease the A-beta 42 relative to 40, and 42 is what drives the amyloid. But they weren't very effective, and they weren't very brain permeable. So Steve and I did the first screen to find non-NSAID next generation gamma secretase modulators, as we named them, 20 years ago. It's taken now 20 years from a company that screened, that was Torrey Pines Therapeutics, at that time with support from ASI. The company went bust when the CEO and board threw the founders under the bus, all too often story, and started a migraine drug. But we took the drugs out, and we continued them in academia with a blueprint grant, and also with funding from the Cure Alzheimer's Fund, and also funding from NIH that Dennis and I had for, how long was that, 20 years?
26 years.
26 years on a program project on exactly this topic. And now, literally this week, in 2 days from now, on my birthday, they will, I'm getting older, we will be finally submitting the IND for our gamma secretase modulator from work that started 20 years ago. And the company that's doing this is Acta Pharmaceuticals. When we finally had an IND ready, the blueprint people said, start a company. Don't put a new chemical entity into a person as an academic. So we started Acta Pharmaceuticals, and then Mass General licensed the IP for the gamma secretase modulators to that company. And that IND will go in now as a collaboration between Acta and MGB. And we're hoping for the best. The phase 1 trial will hopefully start before the end of this year. In the meantime, other companies have made gamma secretase modulators similar to our structures, and are kind of paving the way for what we might expect. And we're seeing nice results there with safety and lowering A beta 42. So we're rooting for those who are competing with us as well because they kind of opened that gate. So I think the GSM could become the statin of Alzheimer's. You have your blood test, like a cholesterol test. You find out you're getting into trouble, and a safe little white pill is something you take to bring it back down. I'm looking forward to that future.
I think GSMs are one small molecule approach. There are others that are being tried, but they make a lot of biological sense. They do a surgical strike on the presenilin mechanism for cutting APP. And not only do they have the advantage of decreasing A beta 42, which is a bad guy, they increase shorter forms of A beta 37, 38, which appear to be anti-amyloidogenic in preclinical studies. So I think this is a great, it's in phase 2 now at Roche with their GSM, and as long as they don't experience off-target side effects, I think this agent should be very good.
But let's not forget clearance of amyloid without monoclonals.
Yeah.
How can we do it? Like microglial cells are the key. Microglial cells are clearing amyloid in your brain, and when they become tired, it's Dr. Jekyll and Mr. Hyde. They become neuroinflammatory. So a lot of the work we're doing in our unit at the McCann Center for Brain Health is coming up with using our organoid models where we can screen known drugs and natural products 100 times faster and cheaper than using mice. And we've done this for the past 8 years. We have many natural products and repurposed drugs that can be used to reprogram microglia, get them to eat more amyloid, and get them to become less neuroinflammatory at the same time. So those trials, we just raised at MGB, we raised $25 million recently, beginning with a $20 million gift, to start a platform clinical trial to reprogram microglia to eat more amyloid and cause less neuroinflammation with known safe FDA drugs that can be repurposed that we found in the dish and natural products. And so we're very excited to get that trial program going at MGB.
And the GSM from Roche, by the way, is being combined in a 2x2 factorial trial analysis with Leqembi. So both something to clear amyloid and something to decrease its production.
Yeah. Great. Okay, we have a grand total of 2 minutes, so we're going to do speed rounds. Going back to trontinemab, just quick question. Regulatory pathway, do you have to show clinical improvement, or can you just file on plaque reduction?
So we've recently put a paper together arguing you should just do it on a biomarker basis. And
Will FDA believe you?
Not yet, not yet. But we think
hope.
Definitely biomarkers should be
But that would be important for the next drug. The next drugs that come through like the GSM, we don't want to have to do a $2 billion 18-month trial. If we safely lower amyloid, the FDA should let us help people.
Exactly.
BIB 080, you talked about this, anti-sense oligonucleotide to tau, lowers intracellular tau, the MABs to tau, I think have failed because they're extracellular.
Right.
Potentially very promising. Your view of that approach?
So my view is that that is very promising and could be combined with an amyloid-lowering agent to get rid of what's outside. Tau is an executor of the Alzheimer process in many ways, and if you didn't get tau buildup or got less of it, you might even tolerate it. And that may be the very reason why we talk about the Achilles heel of the amyloid hypothesis. You have a head full of amyloid and you're not demented, you're not impaired. But it may be that some people don't get as much downstream tau alteration. So lowering tau levels, if it can be done safely, has to be done intrathecally right now, and eventually that might change.
The problem there is you still have to do that early. Amyloid-inducing tauopathy is happening a decade or more before symptoms.
Absolutely.
So if you're going to try to treat tangles or amyloid, it has to be something that's affordable and safe enough to use, not intrathecal. So I think we still have to think about the safest possible alternatives.
But I do think intrathecal will be accepted by patients because intraocular injections for age-related macular degeneration happen every 4 weeks, and these are TV advertised products, and there's millions and millions of people who get them, and it's not pleasant either.
Not exactly democratizing, though, when you have to get it intrathecally.
No, that's the problem.
Preclinical studies, so we touched on this briefly. There's the Trailblazer ALS 3, there's a HEAD 345, so those are looking at amyloid positive patients that have no clinical symptomatology. Will those trials be positive?
So I think they will. It depends on what one calls positive. So one hopes that these patients who are cognitively impaired, that after 4 years of treatment, they will still have relatively little cognitive impairment, and so that'll be the outcome. And their biomarkers will be studied as well, so they're going to have putatively less GFAP in the plasma, less tau, p-tau217 in the plasma as a result. So I don't think all of the patients in the trial, I think there are 1,300 patients in the AHEAD study, will have arrest of their process, but it's given the biology of what we see in treated people with symptomatic disease, it'd be very surprising if there isn't a good signal in the AHEAD study.
But we have to hope the FDA evolves with the field. Look, this is very similar to finally saying if you have a drug that lowers cholesterol, you don't have to show over 5 years you're preventing an MI. This is the same course for amyloid and Alzheimer's. At some point, the FDA has to say if you have safe drugs to lower amyloid, you don't have to do a 5-year prevention trial. You don't have to do a large trial. Lowering amyloid is going to help prevent this disease and help tens of millions of people. We have to get to that stage, just like they did for cholesterol and heart disease. We're on our way, but it's still a long road.
And for those in the audience who are concerned about this particular issue, Azen et al., Alzheimer's and Dementia 2025, is an article that lays out the whole reason why the FDA should think about this.
Right.
Okay, last question, because we're a little bit over. There is one possible way you could democratize Alzheimer's because the GLP-1s are being studied. So the oral semaglutide by Novo Nordisk and the EVOKE trials, supposed to read out phase 3 trials before the end of the year. Will that work?
Well, obesity is a risk factor for Alzheimer's. I think anything you do for obesity is going to help. In our own hands, we don't have direct evidence of GLP-1 agonists actually specifically hitting Alzheimer's pathology, but there's some talk out there that it might be helping reduce cortical thinning in patients who are using it. But I think the jury's really out on that for Alzheimer's.
There's a little bit of preclinical evidence that tau alterations might be benefited by that. Less evidence than that A beta alterations are targeted, but that's
not
okay.
in our hands. Yeah.
We'll see.
Yeah, not yet.
Okay, well, this has been great. Dennis and Rudy, thanks so much. Rudy, you've got a birthday coming up. I was wondering if Dennis might regrade your paper.
Yeah, I would.
Bump it up to an A.